The nature of genes. So, first of all, let me differentiate between gene and genome. So, a gene is a region of the chromosomal DNA that can be transcribed into functional RNA at the correct time and place during development. And as we know, the correct time and place is usually when protein is needed by our body. When it comes to a genome is different. So the genome is the entire complement of genetic material in the chromosome set. When it comes to genome, or when we're saying the genome of an organism, we're referring to the complete set or the chromosomal set of genetic information that is found in the nucleus. We looked at the set of organelles in your first year, and we also know by now that when it comes to organelles such as the mitochondria, they have their own genetic information, and so they have their own genome as well. So the genome is the entire genetic material or chromosome set in a particular organelle. When we're referring, let me say once again, when we're referring to genome of a person, we are referring to the genetic information that is found within the nucleus. And this is why it's so hard to clone a person, because we don't have the genome of all organelles and so on in order to clone an organism. They only know the genetic information that is in the nucleus, and that is why no matter how much tries they have made, the clones are not, the clones, they come close, but they're not entirely identical to the original offspring they're taking in genetic information from. So that's as it relates to genome. Now let's look at the main regions of a gene. So when it comes to genes, remember I said that genes are the regions on the DNA that can be transcribed. So the thing with these regions is they are diverse in their function and in their sizes. But even though they're diverse in so many ways, most of them, they have a certain topographical feature that we can clearly outline, or a physical feature we can clearly outline. And the first feature is the fact that eukaryotic genes, let's use eukaryotic genes specifically, that they contain mysterious regions or segments that are called introns. Now, this segment of DNA, it doesn't contain information for functional gene product such as protein. They are transcribed, however, together with the coding regions, which are called exons. Now, after transcription, this is where they are excised from the initial transcript through splicing, something you should know by now. So, since it doesn't code for anything, what is the purpose of an intron there? So an intron, together with the exon, they're found in association with other regions, for instance, the regulatory regions. Now, all of these topographical features here, they are necessary in order to generate a properly sized transcript at the right time and place. And because of this, we must consider these regions to be part of the overall functional unit. And in other words, we can say that it is part of the gene. Now, if you look here, you're going to see a generalized structure, gene structure in prokaryotes and eukaryotes. Prokaryotes towards the top, eukaryotes at the bottom. If you look at the eukaryotic gene, you have the regulatory region here, which is similar to the eukaryotic genes. Both genes, they have these regulatory regions as well as the termination regions. Now, the difference is that when it comes to the genetic information that actually produces the protein, for prokaryotes, you have coding regions, and for eukaryotes, you have coding and non-coding regions. The non-coding regions, of course, in the light green are called the introns, and the coding regions are called the exons. And so before before a transcript can be used to produce protein, this is why splicing is important. So basically, we have this information being explained here. So we have the coding region in the dark green. We have the non-coding region in the light green. And as you can see also that the coding regions, they're often split by one or more introns. All right, so you have all of that explained here. Moving on to this table now. So this table is representing the relationship between gene size and mRNA size. Let's start off first with the species. So we have a wide range of species here. We have some prokaryotes and we have some eukaryotes. So like Haemophilus, influenza, Metanococcus, Saccharomyces cerevisiae, all of these are prokaryotes. Then we have the eukaryotes towards the bottom, like the chicken and mammals in general. Now let's pay attention to the average gene length. We have them here in kilobase pairs, and we also have the average mRNA length. Now let's look at the prokaryotes. As you can see, the length of the gene is the same as the length of the mRNA. But when it comes down to the eukaryotes, the length of their gene is different from the length of the mRNA. And that is because, as I would have explained just now, when it comes to eukaryotes, we have coding and non-coding regions together in the gene. After it would have been spliced, the coding regions remain, and these are what are used to produce the protein. And so this is why the mRNA would be shorter as compared to the gene. And also, this is why it is the same length for the prokaryotes, because they do not have this coding and non-coding region. This basically describes the table, right? Moving on now, genes are also surrounded by other DNA. So with molecular biology, there's a vast variety of techniques that are known today and that can be used. When you go farther, if you're going to complete your degree, we're going to go into these techniques a little bit more. We're going to learn about all of them, in fact. All right. So majority of them actually involves nucleotide sequencing, and through nucleotide sequencing, and other physical studies, they have shown, these studies have shown that between genes, there is actually DNA, and their function is actually not known. So the size and the nature of this genetic or this unknown genetic information varies in the genome. For instance, in a fungi, it might be a little bit, but in a mammal, it might be huge. And so this unknown genetic information, we say that it's an intergenic region. And this intergenic region, it varies depending on the species. The size, as I mentioned, and the nature, it varies depending on the species. If you look here, we can see the gene topography in 4 organisms. So if you look here, you're going to see we have the bacterium, yeast, Drosophila, and we have human. So if you look, you can see that, if you pay attention, you can see that for the eukaryotes, we have the lighter green areas, we have the deeper green areas. For the prokaryotes, it's just completely dark green. And yet again, this is because of the coding and the non-coding regions. But in addition to that, as I mentioned before, there is other regions of genetic information whose functions are not completely understood. They're unknown to be exact. So if you look here, you can see some examples of intergenic regions between genes for these various organisms. So there is a concept that, um, so the concept is that some places between genes and this non-genetic information, these sequences, they exist quite a distance from the gene, but yet they have the ability to affect the regulation of that gene. Now, because these intergenic regions can affect a gene, we consider it to be part of the functional gene unit. So everything that is there around the gene, it is considered to be a part of the gene unit, not just the part that actually produces the protein. All right. So in many eukaryotes, some of the genetic information or the intergenic regions that are present there, they are actually repetitive. Sorry about that. Yes, they're actually repetitive, consisting of different types of unit throughout the genome, but some of it, these repetitive sequences, some of it they're dispersed, and some of them they're found in contiguous tandem arrays. All right, so they have different patterns that they take. Some of them they're also found in the introns, in that part of the genetic information as well. All right, so the extent of this DNA, it is different as I mentioned before in different species, and there is variation in the repeated numbers within species. So for human, the amount of repeated um, genetic information we have there will vary for human as compared to a fly, as compared to a fungus, and so on. Now, when you move later into your fourth year to be exact, we're going to go into these repeated DNA a little bit more because it is still a mystery. There are several hypotheses about their origin, and we're not going to go more into that for now, but just know that we will revisit this if you're going to be doing the entire degree, and we're going to look at some of these hypotheses and so on. And that brings me to the end of this presentation. And so it is important for you to understand the nature of genes, that they basically contain topographic features, meaning they have coding regions. When specifically the eukaryotic gene, there's coding regions, non-coding regions. We also have intergenic regions, which have their own features. For instance, the fact that the sequences are repetitive and so on. All right, and even though it's not fully understood, it is believed that these regions are important because they help with the regulation of genetic information. And this makes more sense when we think about the fact that each and every cell in our body, they have the same genetic information, but why is it they differ from each other? For instance, why does a nerve cell look different from a blood cell, even though they have the same genetic information? And so this is important when we're going into those concepts. So just to introduce, this course just introduces you into this concept, and I wouldn't go more into it for now.